In the kidney and in many animals that have to deal with high salt environments, the high salt concentrations can result in denaturation of proteins. Other molecules that are present can take the place of sodium and chloride and may help protect proteins from denaturation. Urea has some protective effect, but urea itself, in high concentrations in the kidney and in some animals, also can denature proteins. Other solutes can interact preferentially with urea and or salts and displace the salts from the proteins, protecting proteins from denaturation. When blood moves into the glomerular capillaries and out, urea enters the Bowman's capsule and the proximal convoluted tubule at the same concentration that urea exists in the blood. As water is recovered following solutes, valuable solutes, the urea exists in higher and higher concentrations, and the loop of Henle is not permeable to urea. And so these high concentrations of urea persist. Urea is a waste product, of course. It's a carbon with an NH2 on each side and a double bond oxygen to the carbon. That urea, as a waste product that can denature proteins and cause harm, needs to be excreted. But the countercurrent multiplier, which is moving sodium and chloride out of the ascending loop of Henle and carrying that deep down into the medulla, produces very high sodium and chloride concentrations, which potentially would denature proteins. A urea transporter exists in the collecting duct, which moves urea out into the medulla. That has the effect of reducing the interactions of sodium and chloride with proteins. That in itself is a problem because the urea also interacts with and denatures proteins. Yet another molecule produced by cells in the kidney. TMAO acts to capture urea. Every TMAO molecule tends to associate with two ureas. TMAO is a compatible solute with urea. And in fact, that 2-to-1 ratio of urea to TMAO is very well known as a protective effect. TMAO is one of only many compatible solutes that include amino acids like proline and quite a few other molecules. Often those compatible solutes are compatible with specific other molecules. TMAO and urea is a particularly good example. I'll talk now about how compatible solutes work, and first I need to talk about denaturation of a protein. This is a protein backbone, so a sequence of amino acids all along here. There is a amino acid with a small negative charge, small positive charge, a polar molecule with a negative and a positive region, and that'll lead to hydrogen bonds forming between those two spots. Elsewhere, there's a full negative charge on an amino acid, and there's a full positive charge. If those are close enough, they could fold around and interact with each other, but with sodium and chloride, another possibility is that the negative charge will attract sodiums, and the positive charge will attract chlorides, and there will be no bending here. If we had excess sodium and chloride, now we have a different situation. We'll still have sodium and chloride here, but these two small charges may be targeted with a sodium and a chloride. If there's a sodium here and a chloride here, that hydrogen bond has been disrupted. We have the potential for these to move, denaturation. What about too little sodium and chloride? That can also be a problem. In our same protein strand, we still have the bond here, but if there's no chloride and no sodium, these will be attracted to each other, and we may have a new structure with the interaction between the positive and the negative charge, and again, denaturation. So all species have proteins that have evolved with a specific concentration of sodium and chloride, and if it gets too far from that, they can be denatured. Now, urea also interacts with proteins and can do the same thing. Urea, with its NH2 on each side, has a tendency to attract a positive charge, so a urea can interact with a particular target molecule, disrupt the bond. Again, denaturation, in this case from urea. And that's one of the reasons why high urea concentrations that result from kidney failure are toxic. But in the presence of TMAO, the result is different. In the presence of TMAO with normal sodium and chloride concentrations, each TMAO can interact with two ureas. Now, these are not fixed bonds, but the ureas tend to associate with TMAO, and when they do, they are not free to disrupt the bonds of our protein. TMAO is protective, particularly for urea, and is a compatible solute. Finally, I should mention that there are ongoing debates about exactly how this protective effect works. Some of the debates suggest that instead of interacting with urea, TMAO helps to structure water. The increase in structure of water helps to protect proteins, and that may also be some of the protective effect of compatible solutes.